Good evening. I'm Gina Sapiro. I'm the Dean of the College of Arts and Sciences, and I'm here to welcome you to our first Discoveries program now of this part of the academic year. We are entering the 5th year of this series, and it remains a cornerstone of the philosophy of the College of Arts and Sciences to extend the learning community to the alumni and friends of Boston University as a whole. The College of Arts and Sciences represents the liberal arts and liberal learning, and this is a very important part of our philosophy that one of our gifts to the university community as a whole, including all of our faculty, our staff, our students, and alumni, is to bring here a series of lectures and presentations that stretch across the large variety of the fields, from the arts to the humanities to the social sciences and sciences, on the premise that for all of us, what we all need to enjoy life and to do well in life is to build a broad and deep platform of knowledge that allows us to continue to learn throughout our lives. And so we're delighted to be able to do this and to welcome many of our alumni here who are doing exactly that, enjoying learning throughout their lives. It is 54 degrees outside, and it's not the very beginning of December, but it is early December. This is New England, it's Boston. Once upon a time, we didn't expect it to be 54 degrees on a date like this, or if it was, we'd say, my, how nice this is. I don't have to have my heavy coat and it feels good. But we don't really notice it very much anymore. I came over and I thought, maybe I'll wear a coat, maybe I won't. It's December, 54 is just fine. And with that, I am very pleased to introduce our speaker for this evening, Pamela Templer, who is an associate professor of biology in the College of Arts and Sciences. She is a graduate of the University of USC Southern Santa Cruz, the University of California at Santa Cruz, and Cornell University. Pam Templer has been with BU since 2007. She and her lab focus on ecosystem ecology and the impacts of human activities such as fossil fuel combustion, human-induced climate changes and land use, and the effect on forest ecosystems. So any of you who don't want to hear about human-induced change on the climate, now is the time to go. She and her team currently work in the forests of the Northeastern U.S., redwood forests in California, and tropical forests of Mexico and Puerto Rico. She works with undergraduate, graduate, and postdoc students in her lab, and in addition to her research, she also teaches and is well admired for her teaching in ecology, the biology of global change, and forest ecology. So I'm very pleased to introduce Pam Templer. She's going to give her talk and then afterward entertain questions. When you ask your questions later, we'll have some roving mics so that you can be recorded. And now we'll listen to her presentation on Out in the Cold, Effects of Winter Climate Change on Northern Hardwood Forests. Pam.
Great, thank you very much, Dean Sapiro, and thanks all of you for coming tonight. It's so great to see you, and it's really nice to see some of the students from my class, so thanks for coming. As Dean Sapiro mentioned, I'm going to be talking to you tonight about winter climate change and the many facets of it, but just to give you a brief overview, I'm sure many of you are aware, but in case you're not, forests around the world are being impacted by a number of things that you and I are doing, and so climate change is just one of them. Can you hear me if I look that way? Climate change is just one of them, but there's also land use change such as deforestation, atmospheric deposition of pollutants. So we all live in a city here, so we're big hot spots of nitrogen and sulfur and heavy metal deposition, as well as introduced pests that have made their way from other places that are really changing the species composition of forests around the world. Well, today I'm going to be talking to you specifically about climate change and mostly focus on what's happening, as was just mentioned, here now, so during the winter time. This also might be familiar to some of you. This is known as the Keeling Plot, and this is basically a plot showing you the increase in carbon dioxide concentrations that have been occurring in our atmosphere since the Industrial Revolution, but shown here is since the 1950s as measured in Hawaii, one of the cleanest places in the earth, and showing this quite distinct signal that CO2 concentrations are in fact increasing in our atmosphere. And so what is known is that carbon dioxide is a greenhouse gas, and it's contributing to climate change around our planet. And here in the Northeastern United States, we're no exception to changes in climate that are happening. So what I'm showing you here are mean annual air temperatures for the Northeastern United States. So I'm showing you average temperature on the y-axis. This is in Fahrenheit, with year on the x-axis. And as we all know, there's a lot of interannual variability. You can see the ups and downs. But the general trend here in the Northeast, just like many other places around the world, is that our annual temperatures are going up. Hence, today it's 50 degrees rather than 20 degrees, and we're not in the middle of a snowstorm. Well, with this increase in air temperature has also come a whole bunch of other changes that are cascading throughout different ecosystems around the world. So what I'm showing you here is data for maximum snow depth. So what is that? This is basically a measure of how thick the snowpack is in the Northeastern United States. So I'm showing you data that's coming from Hubbard Brook, which is in the White Mountains of New Hampshire. They have some of the longest running data sets for different environmental variables of the entire world. This is the location where acid rain was first discovered in North America, and they're really well respected for the fact that they take data and they continue to take data for long, long periods of time that allow us to basically detect changes over these periods of time. So this data was collected by the U.S. Forest Service, and as I said, it measures maximum snow depth. As you can see here, this is a Mount Rose snow core, where you just literally stick it in the snow, pull it back out, and measure how deep the snowpack is. And you can see again, just like air temperature, snow depth has been changing over time. Although there is quite a bit of interannual variability, at the two elevations this data was taken from, you can see that over time, the amount of snow that is on the ground is decreasing. In fact, over this about 50-year period, the number of days that snow is covering the ground has been reduced by about 22 days per year. So no surprise, we're in the middle of December right now, and we have no snow on the ground here in Boston. And that's happening up in the northern forest as well. And so what does this mean? What does this mean for all of us? So I love this quote. Inside a snowflake, like the one on your sleeve, there happened a story you must see to believe. And so I think there's a story in here that a lot of us don't necessarily think about. I certainly didn't think about this 10, 20 years ago. But I think the changes in the snowpack are having a big impact on not only forests, but you and I in terms of our daily lives. So I'm going to share with you a few different stories that I got from the news, and just to show you that winter climate matters, but not just in an academic sense. It's affecting a lot of people who make their home here in New England. This comes from the Portsmouth Herald in 2007, and I just want to draw your attention to a little bit of text. The headline here is "Warmer Winters Mean Less Logging." And so for some environmentalists, that might be a good thing. But here in the Northeast, a lot of people do logging in a very responsible way, and they depend on it for their livelihood. And so one thing that's been noted around here is that with less snow, the mud season is actually increasing. So the number of days you just simply can't get traction on unpaved roads is getting a lot more difficult to carry on. So in this article, they say a series of warm winters featuring late freezes and early thaws have extended mud season, hurting loggers in the North Woods. And also, if you base it on conservative estimates of global warming, so using scenarios with very relatively small changes in temperature in the future, we might expect an increase of 10 days of mud, so the mud season, over the next century. And if you use more dramatic basically estimates, but are shown to be actually more realistic in terms of the change in climate over the next 100 years or so, we might see a more dramatic increase in the length of the mud season by 60 days over the next century, which is a loss of roughly 20% of the current logging season, which could result in direct economic loss to the New Hampshire forest industry of almost $12 billion over the next century, and total economic loss to New Hampshire of almost $34 billion, this study found. And so people living on the ground are already experiencing a change in the winter climate, and it's having an effect on their livelihoods. It's also having an effect on people who just make their way on paved roads in the Northeast as well. This is a sign showing frost heaves. If you're not familiar with this, basically it's during the winter when you get some snow, but then it melts and the air temperatures get below freezing. Basically, the soil just buckles underneath the pavement, and you end up with really uneven pavement. So even if you're blessed with a paved road, it doesn't mean you have an easy commute. And also, we know that the snowpack is having cascading effects on the ski industry as well. So there's a lot of evidence that the snow on the ground that's not occurring as consistently as it used to is affecting ski operators. And so far, many of them have been able to make do with this. Many are making artificial snow, but that's true mostly for the really big producers, the ones who can afford the artificial snowmakers, which cost a lot of money. And so those folks who used to run really small ski resorts are either resorting to different methods of economic gain, such as summer fun, so having summer camps at their ski resorts, or they're simply having to close shop. But I love this thing, this story here. This is an example, I think, of adaptation. This was a study that was published in a peer-reviewed journal, but was actually, I found this in USA Today. And basically, it's describing the weekend effect, where different ski resorts are taking advantage of the fact that people in the city might not know exactly how much snow is on the ground in the mountains. And so their estimates of snow depth actually get exaggerated on the weekends when people living in the cities might actually go out to the forest to ski. So I thought that was interesting, although this is being counteracted by what's known as the urban effect. And this is basically, if you and I living here in Boston don't see snow on the ground, it's been shown that we're less likely to travel up to the White Mountains in New Hampshire or the Green Mountains in Vermont to go skiing. It's harder to psychologically connect that it's winter if we don't see snow on the ground here. So I think it's going to be really interesting, and there are many sociologists looking at this to try to understand how is human behavior already being affected and how will it change with projected changes in climate in the future. Another thing that's happening is the maple syrup industry, which many of us are very proud of here in New England. It's also being affected by winter climate change. So the tapping season, which normally happens from about February to April, is getting pushed back. So basically, it's getting earlier and earlier. And so what this study by researchers at Cornell suggests is that in the next 100 years, maple sap will flow a month earlier. And the concern is that eventually it might happen as early as December, and then eventually we won't have it at all. Because to have maple syrup, you basically need to have below freezing nights followed by really warm days the next day, and that's exactly when the trees flow. So if we don't get below freezing nights, we don't have trees to tap. And so speaking of trees, I'm going to switch gears a little bit and switch from what's happening to you and I as humans, and now start talking a bit more about what's happening to the forest itself. Because I think it's really important to think about how this affects all of us, not just in terms of economics, which I think is real and very important, but also to think about the biota who are living in these ecosystems. So what we know, and this is probably somewhat counterintuitive to some of you, is that a reduced snowpack, so if you have a winter with less snow, that actually leads to increased soil frost, so freezing soil. And I'll walk you through this. So imagine you have a winter, a typical winter, I should say, of the past here in New England, where snow is covering the ground. The snow acts like a blanket and actually insulates the organisms that are living underneath that snow. So they're cozy in the winter, they're underneath that snowpack. But imagine now you're in a situation where that snow is gone. And so it's no longer insulating the soil and the organisms who make their home beneath it. And so now the soil is exposed to below freezing air temperatures. Now, a question I often get is, well, it's global warming, right? It's going to warm up. So of course the soils are just going to get warmer too. Yes, that might happen in 100, 200, 300 years from now. But in the interim, as long as winter temperatures get below freezing, which we do expect them to be for quite a while, maybe just not as cold as they have been, when you expose the soils to that below freezing air temperature and they're not covered by this insulating layer of snow, we know they're more likely to freeze. And so one thing to think about is that we know temperatures have changed in the past, and we know that temperatures are likely to increase in the future. So what I'm showing you is data that comes from the IPCC, which is the Intergovernmental Panel on Climate Change, and I'm showing you data specifically for North America, and I've boxed in the Northeastern United States. So what's projected for our region is that air temperatures are expected to warm by between 4 and 4.5 degrees Celsius over the next 100 years. And this is expected to be associated with an even smaller winter snowpack than what we're seeing today. And because there will be less snow, we're expecting there to be a greater occurrence of soil freezing. And so this one way to think of this is somewhat as a winter climate change paradox, because we often think, and we often hear about it in the news, that we're experiencing global warming. We don't think about global warming, but colder soils. But that's in fact what we're experiencing right now. So we're going to have, and we're already experiencing, a warmer winter climate, which could result in colder soil temperatures and greater soil frost. And this is important not just here for the northern forest of New England, but also for much of the world, because currently about 50% 50% of the land mass in the northern hemisphere experiences soil frost on some basis throughout the year. So some have permanent frozen ground, so we have permafrost. Some are seasonally frozen, some are just intermittently frozen. And so the organisms that have lived in these areas have been adapted to different conditions, and we're basically changing that either by increasing soil frost in some areas or melting it in others. And so why should we care about soil frost? Well, I would argue that we could damage much of the biota that are living in forest ecosystems because, again, like I said before, all of the organisms that make their home below ground, so whether you're a root connected to a tree, you're a soil microorganism like bacteria or fungi, or you're an arthropod, so you're just an insect living there, you're now suddenly exposed to freezing conditions, which can impact not only your health, but cascade to impact water and air quality as well. And so why do I say that? Again, I'm showing you data that comes from Hubbard Brook. This was a powerful figure that has stimulated a lot of work in my lab, where again, U.S. Forest Service scientists have kept a very careful record of soil chemistry and stream water chemistry there for decades. So what I'm showing you is data for stream concentrations of nitrate from the 1960s up to the 1990s. And this is nitrate concentrations on the y-axis. And the reason, I'm going to show you in a moment all the details why, but nitrate is if you have too much nitrate in your water, it's considered to have poor quality, and I'll explain in a moment. But one thing they noted is that in spring times, when there was a big pulse in nitrate coming out of these watersheds, they seemed to be associated with winters before that had a relatively smaller snowpack and greater soil frost. So in other words, warmer winters, but still below freezing, with less snow covering the ground, which meant the soils were more exposed to the below freezing air temperatures, and they froze. And that's been correlated with greater nitrate leaching, so nitrate moving into nearby waterways the following growing season. And so this is where I'm showing you here, why do we care about this? Why care about nitrogen leaching? Well, here I'm just showing you a healthy forest with many different trees. Nitrogen is an essential nutrient. It's why I'm showing this as a cycle inside the forest. You and I rely on it. Without nitrogen, none of us would be here today. It's in our proteins, it's in our DNA, but that's true for all living organisms, whether you're a plant, a microbe, an animal, you name it. Everybody depends on nitrogen. And so it cycles naturally within ecosystems. The problem is if something disrupts the nitrogen cycle and you get an increase in nitrogen that's basically moving out of that forest, which we call nitrate leaching, it could cascade to have many negative consequences. So when I say leaching, all I'm referring to is nitrate, which is just a compound that's sitting in soil water that suddenly gets flushed out, say with a rainstorm, and moves into nearby waterways. So moves into a stream, which eventually makes it into a river and eventually into our oceans. And we know from many different studies that when you have an increase in nitrate leaching, that it's associated with the release of N2O, which is nitrous oxide, a greenhouse gas. It's associated with reduced forest productivity, acidification of stream water, so it's been shown to lead to fish kills and other mortality of other organisms, eutrophication, so algal blooms, as well as blue baby syndrome. So in some parts of rural America, as well as many developing nations, when you get nitrate concentrations that are too high and a pregnant woman drinks that water, it could basically inhibit the ability of the fetus to get oxygen, and it can end up with a stillbirth. So there's many reasons, all the way from human health up to further changes in climate, because N2O is a greenhouse gas, to changes in forest health, that we should be concerned with why nitrate might be coming out of forests following winters with a lot of soil frost. And so why would a reduced snowpack and increased soil frost lead to nitrogen leaching? Well, when I started working on this, I was looking through the literature, really trying to figure out what's going on here. Why would soil frost lead to increased nitrogen movement into nearby waterways? Fortunately, there had been a handful of studies at the time I started looking at this question that had really focused on the soil microbes, so those bacteria and fungi that were living in the soil. And many people had thought at first, well, we're freezing the soil, we're disrupting bacterial communities, we're lysing open their cells, and basically their guts are coming out in the spring. But that was their hypothesis. They tested it, and it did not turn out to be true. So they couldn't say those microbes are responsible for increased nitrate. But what some of the early researchers did detect is that in winters with a smaller snowpack and increased soil frost, they found greater root mortality, so more roots were dying. And so that got us thinking, because we've been studying biogeochemistry in forests for years, is it possible that if you have a smaller snowpack, you freeze the soils, you're also freezing the roots, that the roots are then not able to take up as much nitrogen? So like I said, nitrogen is an essential nutrient. If they don't take it up, the trees aren't going to be as healthy. So if the roots are impaired because they've been frozen all winter, maybe that's affecting their ability to take up nitrogen. And so just conceptually, to put this out there, what I'm showing you here is our hypothesis is that with snow and no soil frost, we'd expect the roots to be very healthy, and trees take up most of their nitrogen as ammonium and nitrate. So we thought these arrows would be nice and big to indicate that they're taking up a lot of nitrogen. However, in winters with less snow and more soil frost, we'd expect the roots to be impaired, and so the arrows up for uptake would be lower, and their arrows out for either leaching or losses to the atmosphere as greenhouse gases would go up. And so associated with this, this got myself and members of my lab thinking, okay, if the roots are impaired and they're not taking up as much nitrogen, maybe this could be affecting other parts of trees and their functioning in forests. And so we started to think about forest productivity as a whole. So if trees can't get as much nitrogen, which is essential for their health, maybe they're not going to grow as well. And also, if they're not growing as well and they're not getting the nutrients they need, maybe they're not able to take up as much carbon. And so carbon enters trees during the process of photosynthesis. It's a really important process. Remember, early in the talk, I showed you the Keeling Plot for atmospheric CO2 concentrations. Trees are a really important sink for atmospheric CO2. And so right now, our forests here in the Northeast are considered a net sink. And so all that means is the total amount of carbon coming into our forest during photosynthesis is greater than the CO2 that's leaving during respiration. It's like saying you guys get net payment each month, right? If you work, you get a salary. And so the amount of money you're taking in, your gross salary, is hopefully bigger than the amount you're losing due to taxes, right? And so the trees, just like us, respire and they lose CO2. And so one question we were trying to ask is, okay, if forests here in the Northeast are considered a net carbon sink, which is really important for global carbon budgets, and now the trees are being impaired, potentially by soil frost, is it possible they can't take up as much carbon? They're still going to have the respiratory costs. They still have to metabolize different compounds, and they still have to do everything else that's in their machinery, so this arrow out would probably be the same, but if this arrow in gets smaller, that means they then might become a net source of CO2 to the atmosphere. And so our hypothesis going into this part was overwinter root mortality might result in a later recovery of springtime canopy carbon fixation. So what do I mean by that? We live here in a deciduous forest, whereas we all know right now the trees have dropped their leaves, but usually pretty predictably in the spring they leaf out, and that's when you could measure them taking up the CO2. So our hypothesis going into this is that if the roots are damaged in winter, maybe they can't take up as much carbon early in the following growing season. And so if we're now considered net carbon sinks, it's possible that future carbon uptake could change due to changes in soil frost. And so for the rest of my talk, what I'm going to do is share with you some of the hypotheses we've been testing in our lab, and then share with you a brand new experiment that we just started this summer. And so for the hypotheses that I'm going to share with you today, the first is that a reduced snowpack and increased soil frost results in decreased microbial activity and trace glass fluxes from soils during snowmelt, which I'll come back to, decreased root growth and nutrient uptake, sap flow and canopy uptake of carbon, as well as the abundance and diversity of soil arthropods. So to share with you this work, as Dean Shapiro mentioned before, we've been doing this work here in New England, focusing mostly at Hubbard Brook, which is in the White Mountains of New Hampshire, as well as in Harvard Forest, which is in central Massachusetts. At Hubbard Brook, we work with the dominant tree species there, including sugar maple and American beech trees. And here in Massachusetts, we work with the dominant tree species, including red maple and red oak. Okay? And so for the first study, this was done by a graduate student in my lab, Andy Ryman. He was really interested in trying to understand what was going on specifically during the snowmelt period. And the reason he wanted to focus first on the snowmelt period and not the entire growing season is that here in New England, as I mentioned before, the snowpack accumulates in winter, and then it typically melts in a very short period. And so this is what it might look like in a typical winter, if we get snow this winter. And then all of this snow will typically melt within about a week's time as the temperatures warm up in the spring. And so we know from past work that solute, so things like nutrients or dissolved organic matter that's locked up in that snow is suddenly flushed out when the snow melts in the spring. And this is associated with an increase in soil temperatures, because remember, the soils weren't freezing before because they were covered by snow, but even so, they tend to increase as soon as the snow melts because the air temperatures have gotten above freezing. It's known that this period can stimulate microbial biomass production, so the bacteria and fungi that are living in the soil suddenly start to really grow quickly. And we know that trace gas fluxes can increase as well. And trace gas fluxes are just a fancy term for compounds that are things like greenhouse gases like nitrous oxide, methane, and carbon dioxide. And so one thing we didn't know before this particular study is, what is the legacy effect of soil frost on compounds that are leaving the forest during this very specific period, the snowmelt period, whether it's trace gas fluxes or leaching down through the soil profile. And so for this experiment, what Andy did in my lab, he's a PhD student, as I mentioned, is he took soil cores up at Hubbard Brook. So if you're not familiar with what that is, I'll show you. He took soils from the top 5 centimeters of the soil, and this is the really yummy stuff in the soil. This is where all the activity is happening. So you can take a soil core down to, 4 meters in some cases around here, though often not because you'll hit rock. But most of the really active biota are right here in the top 5 to 10 centimeters. So we took this soil and he put it in these PVC pipes and then literally just stuck them in the ground and let them stay there for about 5 months. And the idea being just to put them back in the ground, let them equilibrate out throughout the entire winter. And then he brought them back to the lab to do a microcosm experiment and to look at the microbial response to changes in soil freezing. So this is Andy here. So what we did is we had these giant chambers with different temperatures, and what we could do is take soils that were in these PVC cores and either leave them unfrozen the entire time in the lab, a mild soil frost, so just about -2 degrees Celsius, or really severe soil frost to see what's going on when you really freeze these soils. We let them equilibrate at these different temperatures for a week, and then we brought them all up to above freezing temperatures to basically stimulate a big snowmelt event. And at the beginning of this time, we added about 850 grams of snow to each of them. We measured this out to make sure it was equivalent, and then we basically watched as the snow melted, and then very carefully, this is Andy putting snow in the cores, measured trace gas fluxes coming out of the soil, as well as leaching losses out the bottom. And so what did he find? I'll walk you through all this data. So basically, first I'm going to show you the data for carbon, and then I'll show you the data for nitrogen. Just to walk you through this, remember CO2, carbon dioxide, is a greenhouse gas, so we're very concerned with any process that leads to either increases or decreases in the flux of CO2 coming out of soils. And nitrogen, of course, N2O is another greenhouse gas we're concerned with. And in terms of leachate, remember, we're concerned with nitrogen that gets into our groundwater and into our spring water and drinking water. And so what he found, and I should say throughout my talk, different letters signify statistically significant differences. And so what he found is that in the temperature treatment that was warmest, basically the soils did not freeze at all in these soils. He found the greatest flux of total carbon as CO2 and leachate, and that was true for nitrogen as well. So the warmest soils had the greatest losses of both carbon as well as nitrogen. And I should say this was not what we were expecting. It was actually just the opposite, because remember, we thought if you freeze the soils to simulate a smaller snowpack, we thought the losses would actually be higher. And so what these data suggest is that at least in really severe winters where you take out the snow and the soils get really, really cold, -15 degrees C, that we might actually offset some of the losses of carbon and nitrogen, but only for a very short amount of time during that snowmelt period. And so for the rest of my talk, what I'm going to focus on now are our large-scale experiments we've been conducting, and mostly today I'm going to focus on the trees. Andy has continued to do a lot of measurements of soil respiration and carbon and N2O losses from the soils, but for tonight's talk, I'm going to focus on these big guys here and talk to you about how they're being impacted by changes in the snowpack. And so what we've been doing now over the last 5 years is we've had 2 ongoing experiments, one at Hubbard Brook and one at Harvard Forest. And in each of these, we call it our snow removal experiments, where we have reference plots that maintain a natural snowpack throughout the winter. And then we have snow removal plots where I keep graduate students in really good shape. Basically, I don't know how I find them, but they love doing it. For the first 4 to 6 weeks of winter, basically, the group of students in my lab and people we hire from outside literally get themselves up to Hubbard, Brook, or Harvard Forest, get their shovels, and literally shovel. And I have to admit, I have not worked as hard on this as they have, and what I have done is really hard work. And so we have a new experiment now I'm going to share with you where we're actually warming up a forest, and the infrastructure is just amazing. This is so elegant because what could break here, right? You break your shovel, you just go to Home Depot and get a new one. The only thing I worry about is their backs more than anything else. But so far, they've all been okay. But basically, literally within 24 to 48 hours of every snowstorm in December and the beginning of January, they are out there shoveling. And so each of our plots are 13 by 13 meters, and the idea is we want at least 3 to 4 individuals of mature trees at the center of each of our plots. At Hubbard Brook, we have 4 reference and 4 treatment plots. And then at Harvard Forest, we have 3 reference and 3 treatment plots. Okay, and so throughout the winter, once we're done shoveling, we just basically get a team of people out there who on a weekly basis make measurements. And so throughout the entire winter, we're measuring snow depth. So remember that graph I showed you at the beginning with lots of variability, but the trend for lower snowpack over time. We're trying to also measure snowpack in each of our plots so we can document how we're changing it due to our treatment. And then we're also measuring soil frost depth. So what I'm showing you here is this is a PVC pipe that's buried in the ground. And basically, we have a stopper at the top that's connected to clear tubing. And what you do is you fill that clear tubing up with this methylene blue dye that amazingly moves away. I don't know how, but it moves away when the air around it gets frozen. And so it's beautiful because we can then go out on a weekly basis and pull these out of the ground and measure the depth to which the surrounding soil is frozen. Okay, that gives us an indication of the conditions below ground. So what are we finding? We have many of these types of plots. Like I said, we've been doing this for 5 years, but I wanted to give you a representative plot. Other than last winter when we had virtually no snow, this is what every other winter has looked like over the last few years. So I'll walk you through this. This is the winter of 2008-2009 at Hubbard Brook. Everything below 0 is the depth to which the soils froze. Everything above 0 is the depth of the snowpack. Okay? And so the black lines represent the reference plots. The lines with white squares represent our snow removal plots. And for those of you who like statistics, these actually have error bars. You just can't see them because the variability is so low. And so what this is showing you is that in our reference plots, in this year, the snowpack basically just accumulated over time. This was a pretty good snow year, almost 100 centimeters, so almost a meter of snow on the ground. And then basically during the snowmelt period, you can see how it melted. And then in our snow removal plots, you can see that we let some snow come on at the beginning of winter, and then we started to shovel, and then we ended here, and then we let a natural snowpack accumulate for the rest of the winter. In our reference plots, this black line right here, you can see there's virtually no soil frost in those plots. But in the plots where we remove snow for the first few weeks of winter, you can see not only did the soils freeze, but even after we let the snow accumulate the rest of the winter, that soil freezing or that soil frost treatment was maintained for the rest of winter. And then you can see this sudden warming up at the end during this snowmelt period. So this is hopefully to convince you that we did in fact induce a treatment by removing snow just at the beginning of winter. We basically exposed the soils to below freezing air temperatures, and we froze the soils as a result. And so we also have soil temperature sensors throughout our forested plots. And just to give you a sense that not only did we increase soil freezing, but in the middle of winter, we also induced an increase in freeze-thaw events. And so that also has to do with just like with roads buckling in the middle of winter, the soils are doing the same thing. They're not only freezing, but right at the surface, they're actually going through these freeze-thaw cycles. And so this is just a different winter. This is this is spring of 2011. Temperature in Celsius on the y-axis, and you can see our snow removal period here. But I'm showing this data to illustrate that in the reference plots, you just have this nice clean set of data where soil temperature is above freezing throughout winter. And then in those plots where we remove snow for just the beginning of winter, we ended up inducing freeze-thaw events. Okay, so what did this do? So now we know we had a treatment. We know we've affected soil temperature. Now, what effect did that have on the rest of the forest? So what I'm showing you now is data for soil nitrate. To measure soil nitrate, you literally take a ceramic cup known as a lysimeter and you bury it in the soil, and then you leave it out there and you sample it on a weekly basis. And so what we found, this is from 2008 to 2010, and I've shaded here the periods of soil frost. You can see nitrate is on the y-axis, and what you can see is in our reference plots, which are in blue, virtually no nitrate coming out of those soils. So this is a measure of how much nitrate has the potential to get into nearby waterways, because we're measuring the nitrate right below the rooting zone. And you can see in the snow removal plots, we got this really big pulse of nitrate coming out. And so a big question we wanted to ask then is, okay, they found this at the watershed scale just in terms of observation and pure correlation. We've now induced it in an experiment and found this very striking result, that nitrate is increasing in the growing season, so June and July, following a winter that had increased soil frost. So now we said, okay, we have questions we gotta answer even more here. So that's when we decided to focus on root uptake and nutrient uptake to try to figure out if that's being impaired by the soil frost. And so what did we find? What I'm showing you here is data for root biomass. This is just an ingrowth core that we literally just bury in the ground. And so what you do is you get a whole team of people, and you basically take soil out of the ground. You pick out every single root you can see with your eyes, and then you put the soil back in the ground. And so we call that root-free soil. And then basically you stick that in the ground, you let it sit there for about a year, and because this mesh is about the same size, the openings, as roots, you can then collect these a year later and get a measure of the root productivity, so below ground productivity. And so what we found here in this 1 year period is that the root biomass in our reference plots was significantly greater than the root biomass in our snow removal plots. So remember, I had shown you that earlier studies found increased mortality. They were just sampling roots to see who was alive, who was dead, but we're now measuring the actual productivity of the roots that were already there and showing that there's a 59% drop in how many roots are actually growing into these soils. Now, part of this might be explained by root injury. This is work where we measured what's known as electrolyte leakage. So literally think of a root with cells in it, and basically you can take those roots, bring them back to the lab, and measure how many ions actually leak out of those cells. And the more ions that leak out is a measure that the cell membranes are no longer intact, and so the roots are no longer as healthy. And so what we found was between the reference and the snow removal plots, about a 37% increase in electrolyte leakage occurring in the snow removal or plots with increased soil frost. Now, fortunately for these roots at least, they were below the 50% threshold. And why that threshold matters is it's thought that once you get above 50%, where the roots are leaking out more than 50% of their content, that's basically associated with tissue mortality. So what I'm showing you here are roots that are still alive, but it's showing that they've been injured. And so now we wanted to translate that into function. If these roots are being harmed, what impact does that have on their ability to take up nutrients? And so this is a picture of Annie Sochi, my first PhD student who just defended last fall. And what she did in her dissertation for one of her chapters was what she called root tickling, where she would literally take roots and keep them still connected to the trees, which is pretty amazing because most people will just say, chop off those roots, bring them back to the lab, and then do some stuff to them. But what she was doing here is keeping those roots connected to the trees. She would put them in a solution with a known concentration of nitrogen. She'd let them incubate for about an hour or so, and then bring the solution back to the lab and measure the change in nutrient content of that solution and use that as a measure of nutrient uptake by the trees. And so what I'm showing you here first, because this work was so central to our original hypotheses, is she first took data before we even did any snow manipulation. So this is just data from our plots before we started any of our treatments to show you that the responses were similar across our plots. So there wasn't some inherent difference amongst our plots before we even got started. So this is just showing you in 2008, the different months she went out and measured roots. And this is net uptake of nitrogen as ammonium up here and as nitrate down here. And you can see there's no difference among the reference and the snow removal plots because we hadn't done our treatment yet. Okay, now I'm going to show you the good stuff. This is showing you the difference in nitrogen uptake between the reference and snow removal plots after a winter of snow removal, where we, again, only shoveled for about the first 4 weeks of winter, but we induced soil frost throughout the entire season. So this is showing you the spring of 2009. She also sampled out into July and September, but really focused at the beginning of the growing season when we expected the largest results since the roots had just been damaged due to soil frost. And so what I'm showing you is net uptake of nitrogen on the y-axis. So this is ammonium plus nitrate, so their total inorganic nitrogen uptake. And what you can see is that if you compare the reference plots to the snow removal plots, there was about a 36% drop in the total amount of nitrogen taken up by trees in the plots where we remove snow and we induce soil frost. And what does that mean? Well, what we did is a back-of-the-envelope calculation. So we just wanted to know, could that reduction in nitrogen uptake by trees account for the increase of nitrogen coming out of the plots following soil frost? And so what we did is we said, okay, how much nitrogen do trees typically take up? Well, on average, at least at Hubbard Brook, they take up approximately 40 kilograms of nitrogen per hectare per year. Well, if we look at a 36% reduction, that would be about 14 kilograms of nitrogen per hectare per year. We know from our lysimeter data, so the leaching data, that the forest nitrogen loss due to soil frost is roughly 14 to 25 kilograms per hectare per year. From that past study I showed you before with root mortality, they found that you can account for about 5 kilograms of nitrogen per hectare per year just due to necromass, so basically just dead roots that are not doing anything anymore. And so if you take that into account, the nitrogen loss as leachate alone due to soil frost is about 9 to 20 kilograms per hectare per year, which basically overlaps with our estimate of the reduction in nitrogen uptake by trees. So we can't prove it, but the evidence suggests that that reduction in roots, so the injury that's being caused to roots, their diminished ability to take up nitrogen, is having a large impact on the ability of these forests to retain nitrogen. And again, we're concerned because nitrogen has all those cascading effects on water quality. Okay? What about other measures of tree health? Sap flow is just a fancy word for water uptake, and then, of course, canopy uptake of carbon, which I mentioned earlier. Now, to measure sap flow, what we do is we basically treat our trees like patients, and we hook them up to syringes. Basically, I'm showing you a close-up up here. Imagine this is a tree. This is the bark of a tree. And we literally put in syringe needles that are hooked up to wires, and we measure how much water is moving up through the tree. So what this is over here is just a cartoon schematic of the cross-section of a tree. So what we do is we put in a sensor here and a sensor here about 10 centimeters apart. And so trees get their water from the soil. And so what we do is we heat this upper probe on a continuous basis. And so we measure the temperature difference between these probes. And so if that temperature difference is really high, that actually means the tree's not taking up very much water. But if their tree is taking up a lot of water, then the heat actually gets dissipated with the water going up the tree, and the temperature difference is not very big. And so we monitor this on a continuous basis throughout the growing season. So as long as trees have leaves, they're taking up water. And so we had 4 sensors per tree. For the data I'm going to show you here from Harvard Forest, we had 2 red oaks and 2 red maples per plot, and again, we had 6 plots total. And so just to show you a close-up, these are the 2 sensors on one tree that are hooked up. And then we also made measures. Remember I said we talked about carbon uptake by the trees. This is a Lycor, where we literally clamp it down on a leaf and measure how much CO2 the tree is taking up. Okay? And so what we first wanted to do before measuring the sap flow and the canopy carbon uptake is to figure out if shoot growth of trees is actually being affected, because that's a direct corollary to how much water and carbon the trees can take up. So what we've shown here is that in our reference and snow removal plots, the amount of leaves that have the ability to control water or carbon uptake is actually, the length of those shoots is actually decreasing in our snow removal plots. And so let me show you some of the sap flow data. Again, sap flow, just a fancy term people use for water uptake. So what I'm showing you here on the y-axis is grams of water per meter squared of sapwood for a 30-minute period, which is a standard way to measure it. I'm showing you data here for our red maple trees at Harvard Forest. Up here is the average values for our reference plots, and down here are our snow removal plots. Probably what you're noticing right away is this big gap in the data. For some reason, Harvard Forest gets an amazing number of thunderstorms in the middle of summer, way more than Hubbard Brook. And so that often basically tears out our power, and then we don't have the ability to make measurements. So sometimes we do gap filling, but only if you have very short periods of no data. But certainly when it's periods like this for a couple of weeks, we don't attempt to gap fill. So that's why it's empty here. But luckily it's empty for all of our plots. What you can see here is this ramp up in the beginning of the growing season. So if you're not familiar with Julian Day, this is just days of the year. So number 1 is January 1st, 365 is December 31st. And so the beginning of the growing season in this particular year was about May 18th. And then I'm showing you data for about the first 6 weeks of this particular year. And so each of these peaks represents a 24-hour period. And so trees are amazingly predictable. They wake up every day, and in a sense, they go back to sleep. And so each peak basically represents the day that, when they're awake and when they go back to sleep. And so trees, if you're not familiar, have stomates on the bottom of their leaves. And as they open and close them, that basically pulls water up through the roots and then out through their leaves. And so what I'm showing you here is this early leaf expansion. So you can think about it next spring as the trees put on their leaves. Not only are those leaves getting bigger, but all the fluxes going through that tree are getting bigger too. So water uptake, most likely nutrient uptake, carbon uptake, all sorts of things. So that's what I'm showing you right here. But here, again, we're trying to understand what is the impact of a smaller snowpack and increased soil frost. So I'm showing you the same magnitude on both of these figures, so 4,000 up here, 4,000 here. And you can see just from the data alone that if you look in the snow removal plots, the amount of water being taken up at the beginning of the growing season is substantially decreased in these red maple trees compared to that of the red maple trees. And so because it's somewhat difficult to think about this on a daily basis, I'm now going to condense this data into the first 29 days of the growing season. And so like I said, I showed you red maple data in the last figure, but now what I'm showing you is data for both red maple and red oak trees. So on the y-axis here is sap flow, just a measure of water uptake by these trees. The black bars represent the reference plots. The hatched bars represents the snow removal plots. So again, different letters mean significant differences. So you can see for the red oak trees, no statistically significant difference among the reference and snow removal plots. But the red maple trees, you can see this drop between the reference and snow removal plots. It's not statistically significant, p-value is 0.1, but we've continued to monitor this, and it appears to be a consistent trend over time, that especially with red maple trees, they have a drop at the beginning of the growing season. And so this is just for sap flow, so this is just a measure of water uptake, and if we calculate, it's about a 23% drop. Now, knowing that trees have stomates and they control how much water they take up by opening and closing their stomates, we also know that opening and closing their stomates is also how trees get carbon dioxide for photosynthesis. So using a lot of different calculations based on sap flow and leaf area index and other measures of tree biomass, we're able to convert these measures of water uptake into canopy carbon uptake by the trees. And so that's what I'm showing you here. This is carbon uptake in micromole CO2 per meter squared ground per second. And again, what you can see, not surprisingly, because it's proportional, is that in red oak, there's no difference. But in red maple, this big trend that you get a reduction in carbon uptake by the trees in spring. And so remember, this is really important because we currently consider these forests to be net carbon sinks. And what this suggests is that the damage to roots and the damage or diminished ability of these trees to take up water and nitrogen is having a cascading effect on their ability to take up carbon as well. And this corresponds to a 23% drop as well. And so what did we find with sap flow and canopy carbon uptake? Interestingly, we found in general the magnitude of uptake of water was greater, for water and canopy carbon uptake, was greater for the red maple than the red oak trees, which shows that not all species are the same. We found that a smaller snowpack and increased soil frost had no impact on red oak, which was surprising to us, but decreased it in red maple. And so what this suggests is that tree species matter. We can't just consider our forest as a black box, but knowing what species is there in terms of how projected changes in climate might occur will really help us to understand how different trees will respond in the future. So now I'm going to switch gears slightly. For the last part of this part of my talk is just switch gears and talk about soil arthropods. I should say I am not an entomologist. I always like to qualify this. But to show that undergrads can have a huge impact on the direction of one's lab, I'm sure many of you had an impact on labs as well and other fields, is an undergrad in my lab. I have a picture in the next slide. Andrew Schiller came to me in my forest ecology class and said, "Professor Templer, I really like bugs. I want to study them in your plots." And I said, "Andrew, I don't know anything about bugs. I know some, but certainly not enough to guide you very well." And the beauty of it was basically Andrew ended up getting co-advised by me and my colleague Tom Kuntz, who knows a lot about insects. And so it was a wonderful bridging of two different people in different fields who then co-mentored the student, and it really turned out to be a very productive collaboration. And the reason that this still relates to what we'd already been doing, although it was very complementary and different, is that soil arthropods are really important for ecosystem function. They enhance rates of decomposition, they alter microbial communities, and just the presence of them in soils has been shown to enhance rates of plant nutrient uptake and growth. And so what Andrew did is he took litter, so literally leaves from the ground in our different plots, the reference and snow removal plots. He brought them back to the lab in 2009 and 2010, and he put them under these funnels with heating lamps. So basically it causes the insects to move down into these cups, and then he can measure their abundance as well as diversity. And so again, the question was, if you make the soils much colder in winter, does that affect the ability of soil insects to grow and to live? And so this is Andrew down here. So I'm just going to show you some representative samples we had. We had many different orders that he looked at. But just to give you a sense, there's spiders here, pseudoscorpions here, and then Hymenoptera here. The black bars represent our reference plots, and the gray ones represent the snow removal plots. And in all of these cases, we found a significant drop where when we took away the snow and we induced soil frost, we reduced the abundance of each of these groups. And then when we looked at all of the orders of insects together, what we found is that the richness of the orders as well as the overall diversity declined as well. So richness is just basically the number of orders present. So what he found statistically significant in both 2009 and 2010, when you compared the reference to the snow removal plots, there was a significant drop in both richness as well as diversity of these plots, which I thought was really interesting. I didn't know what to expect going into this. I thought maybe they could just go deeper in the soil or they just move out of our plots. But what it shows is that the arthropods that are there, not only are the trees being affected, but the insects that make their home in soils in winter are being affected as well. And so what did we find in these series of studies so far? We found that winter climate change has a big impact on forest ecosystems. I'd say more than we were initially expecting to find. We found that a smaller snowpack and increased soil frost reduces microbial activity and trace gas fluxes during snowmelt. It increases nitrate leaching to nearby waterways. So if we care about forest health or acidification of streams or our drinking water quality, we really need to be paying attention to this. It decreases root productivity and health, as well as rates of nitrogen, water, and carbon uptake by some tree species. And it also decreases arthropod abundance and diversity. And so, as I said at the beginning of my talk, seasonally frozen ground is a really big component of our hemisphere. And so if we're changing winter climate, not only are we affecting forests, but we're affecting the people who make their home in these areas. But one question I often get that I've gotten in the past with our work is, wait, you're just changing one thing. You're changing snow. Isn't the air temperature going up too? But you're not getting the full picture. And I would say, yes, you're right. But you have to start with one thing to tease out the effects. And so this question just kept coming, and I kept thinking about it, and finally decided, no, people are right. We really do need to be thinking about not just winter, but what's happening throughout the year. And so what we've been doing recently, we just started it this summer, is now trying to link not just what's happening in winter in terms of winter climate change, but also what's happening when we increase temperatures throughout the growing season. So we're basically now looking at the interactive effects of soil frost in winter and warmer growing seasons on both nutrient uptake by trees as well as carbon sequestration. And so our hypothesis going into this is a picture from just this fall at Hubbard Brook. Our hypothesis is that warmer annual temperatures, so basically global warming, results in higher nutrient uptake and capacity by roots and increased carbon sequestration by trees, but this response is offset by the negative effect of soil frost in winter, which I've already discussed with you. So to try to get at this change in both winter and the growing season, we started a new warming experiment at Hubbard Brook. This is where I miss the shovels. So for the last about 4 or 5 months, we've been working with an engineer, and we've now laid down, you can't see it, but underneath the litter here is almost 4 kilometers of heating cable that's buried to about 10 centimeters depth. And so we've got about, you can only see 2 plots here, but we have 6 plots total. And the idea is that we have 2 plots that are just going to serve as our reference plots. We have 2 plots that we're going to warm up throughout the year and say, okay, what happens to these forests in a warmer world? So in like 200 years when the soils never freeze. And then what happens when we look at the net effect of colder soils in winter with warmer soils in the growing season. And so we're making all sorts of measurements, basically a lot of the things we've already been measuring, but now trying to understand how changes across seasons will affect them. And we're also doing a new experiment, which I'm really excited about, which is a sapling experiment, because we couldn't do this second experiment with mature trees. We're doing this with sugar maple saplings, and we're not just inducing freezing or inducing warming, but we're actually going to change the number of freeze-thaw cycles throughout the winter. So some saplings will experience 2 freeze-thaw cycles, some 4, some 6, some 8. And the ideas we want to get at, is there a threshold number of freeze-thaw events in winter that really affect these trees, and what number of freeze-thaw cycles does that? And then we'll make a bunch of measurements again in the forest, and then we'll be able to feed this into an ecosystem scale model that's already calibrated to the northern forest. And then we can make projections into the future saying, okay, we know how much climate's going to change. How will that affect the health and ability of these trees to take up nutrients and water and carbon and everything else? So we're excited about it. Last and probably most importantly is I have to thank the amazing number of people that I've been able to work with here at BU, and I love them all. I've been blessed with a huge number of undergraduates who work in my lab as either UROP students. Many of you are probably familiar with that wonderful program, Undergraduate Research Opportunities Program. I've got work study students, students doing research for credit, as well as work for distinction, so honors senior thesis. I've also been blessed with wonderful high school students from both the Boston and Brookline public schools, and great PhD students, as well as a postdoc from Germany, and wonderful technicians. I'd really like to acknowledge my funding sources as well, and thank all of you for spending your evening here tonight, and would be more than happy to take questions.
So I'm wondering, having heard all of this, can you describe an optimal winter for the Northeast?
Mm-hmm.
Maybe in terms of when the first snowfall takes place, average temperature ranges?
That's a great question. I would say, what's optimal? That's a hard word. I'd say these forests for, decades, centuries have experienced a pretty solid snowpack from December to April. So I'm talking Hubbard Brook, so New Hampshire, White Mountains. We've always been a little bit buffered here in Boston because we're at the coast and we're an urban area. But I'd say for the northern forests, a typical winter would have looked like about 4 to 5 months of snow on the ground, and the soils would not have frozen. And if they did freeze, it would be about 1 centimeter deep, not 10, 15, 20 centimeters.
Have you, I see you're starting to think about the 5-degree C change, and I'm wondering where that thought process is going, and what research you see coming to address the ability for these trees to take up carbon and their reduction. We're losing forests, and then if we're losing the quality of our forests, where does that lead us?
So do you mean in particular with this experiment, or even past that?
Past that.
Yeah.
Because I think, you're bringing up a lot of really important questions. So where does that take us?
That's a great question. I think what we need to be able to do is quantitatively project how these forests are going to change in the future. There's been a bunch, some of you might be familiar with this, experiments looking at climate change. So people warm up a forest here, they warm up a grassland there. But it's really hard to connect that to the rest of the world. And so I think if we could start to incorporate these responses into models and then extrapolate to even broader regions, I think we're going to get a better handle on how the world is going to change. And so we need to connect this kind of data with things like range shifts. So I mentioned sugar maple in terms of maple syrup decline, but we also know sugar maples don't do well in really warm temperatures. So if it might be that in the next 100 years, their ability to take up nutrients and carbon goes down, but we might not have sugar maple here in 200 years. So I think what we really need to do is not just say, what are the current tree species going to do, but what about the future stock of tree species we get around here? Hi.
Thank you for doing this. It was very good.
Thank you.
My question is, there are some winters, like last winter, where I didn't write out a check for the snowplow
Absolutely.
man at all. There are other winters where I think I sent his children to BU.
Yep.
So how does that all average out? And have you ever done it when we've had 100 inches of snow?
Yes, so basically the first winter,
And I
yeah.
realize research is over a time frame.
No, it's a great question because it's variability and it's natural that we have, and that's why I feel blessed that we've been able to continue this work. Most funding cycles occur over 3 years and then you're done. Fortunately, with things like shoveling, we can just keep it going. Even though the funding for that first project is done, I have great graduate students who want to keep this going, and so shoveling isn't very expensive, and so they're going to just keep going. But with the warming, it's really expensive. So we have funding for 5 years. We'll see what happens after that. But in terms of your question with the snowpack, so that first winter, we got really lucky. There was 100 centimeters of snow, and I'd say that gives us the best treatment because we're taking it away. Whereas this last winter was amazing. We had no snow, I shouldn't say no, very little snow. And one of my PhD students, Patrick, said, I'm going to go shovel anyway. We got one storm at Hubbard Brook last December. And amazingly, that one storm he shoveled with the team, we actually got a treatment. But instead of having no frost in the reference and then deep frost in the snow removal, it just moved everything down. So even the reference plots froze to about 10 centimeters, but the snow removal plots froze to about 30. And so I think having these long-term experiments are really valuable to capture what you're saying, the variability.
Take one more. One more? Good.
Are you seeing the same results, or obviously not of your experiment, but in Northern Europe, is the snowpack dropping there as well?
Yes, and actually there are some people in Bayreuth, Germany, who are doing very similar experiments just in terms of snow removal, but they're focused on grassland experiments. And so they're finding very similar things, that when you get a smaller snowpack, you actually induce soil frost. So we've been talking about actually trying to do some synergies, so working together to ask some of the same questions. They have the same types of curiosity about how a species shift's going to impact things, but they're mostly in grassland ecosystems.
Thank you. Well, I want to thank Pam very much on behalf of my sugar maples up in New Hampshire, who had a 7-day sugaring season last year. Not kidding, it all got zapped, and luckily for us, it's just a hobby, but for a lot of my neighbors, it's for real. And so there were a lot of people who lost quite a bit. Let me just point something out that's here on this wonderful last slide, and that is what research at Boston University involves. And this, I know, is not just the College of Arts and Sciences, but every school and college we have at BU where people are doing research. And that is the style of education and research that we do at BU involves a wonderful kind of apprenticeship of our faculty, our experts, along with their PhD students and their master's students and hordes of undergraduates. When some of us went to college many years ago, only the top of the honors undergraduate students might get a chance to work side by side with a faculty member doing original research. Today, we believe that the students of tomorrow will need to know how to do original research. They'll need to know how to do discovery. And the only way we can do that is not by giving them textbooks and having them read about research, but having them work side by side with wonderful faculty like Pam Templer and her first-rate PhD students. So these students are running around with their shovels and their instruments and everything else, learning about the excitement of discovery, learning about the problems of trial and error, learning about discovery. So I'd like to pick up another thread. I would be remiss if I didn't say this. If this winter you don't have to put your snow removal people's children through BU, let me suggest that you take that support instead and help us to bring these undergraduates and these graduate students to be able to do this research. It's very expensive for us to do this, but it is just essential to support undergraduate and graduate research and the ability of our faculty to work with them. So thank you very much for that setup line. I heard you. So thank you all to all the alumni and students and friends who are in attendance here. Let me just set up the next talk. We make sure that in each year we do very, very different topics. So we hope that we will see you at our next Discoveries program when we are going to welcome experts from across the country as we celebrate the 50th anniversary of Betty Friedan's publication, The Feminine Mystique. On February 12th, 2013, that week is the 50th anniversary of the Feminine Mystique, which had a major impact on the way women thought about their lives, on the origins of the women's movement. And we're really delighted that we're going to have for that not one speaker, but a panel, all of whom have a relationship to Boston University. I think it's 3 of them are very distinguished scholars from around the country who were undergraduates at Boston University in the 1950s and 1960s. And we also have members of the faculty here, so they will be doing a panel on the significance of that 50th anniversary, looking back both at that book in its time, but also asking what's changed since that time. So I hope you'll look forward to that, and we'll be giving you that date later, but February 12th, 2013, the next discoveries. In between that, I wish you very good holidays and a very happy new year. And for those of you who ski, I hope you have snow. And please join us now for a light reception of coffee and dessert in the atrium, located just out there where you can have some more conversation. So thank you very much for coming this evening.